show a molecular QTL browser that we've developed in part of dbGAP's team. This was originally supposed to be for GTEx specifically, but ended up we added some other prototype studies initially, and then Framingham Heart Study had a very, very large expression QTL and other microRNA QTLs that they wanted to add, so we've included those, and those actually swamp out the database. But I can kind of show you what we've developed as this prototype and maybe get some feedback from you. That would be nice. The way this browser works is that we have billions of p-values, which are mostly eQTLs, but also we have MES-QTLs and miRNA QTLs. And what we're doing for the eQTLs, we're looking at genomic position and how that affects expression within a gene, usually measured by a probe within that gene. I don't know if there's any questions about what an eQTL is.
Do you want to just maybe a brief explanation would be great, just to make sure that everybody's on the same page before we go into more detail.
Okay, so in a single person, you can do two assays. One is a genomic sequence or GWAS chip array, and each position, each SNP, each variation can be measured against another assay, which is an assay that measures the expression of a gene, and the correlation between the genotype and the expression of that gene in the other assay can be measured, and you can assign a p-value to that measurement. Hopefully that explains kind of what we're talking about. So this database it's set up with a kind of a little browser that we've created, and in this browser you have some options to filter, and those are on the left. So you can filter by the genomic location that was measured on that person, on these people, and then you can search. You can kind of filter the results by a specific RS number, or by a chromosome, or a specific gene if you're interested in a gene. And then on the molecular phenotype, this is the probe that's measuring the expression. You can filter by the probe IDs that were used in the assay, the chromosome that probe is hitting, or the gene that probe's hitting. And then you can filter also on the type of QTL, which might be expression QTL, MES QTL, or miRNA QTL. And then you can also filter by the p-value, and we did this in a log, so you can measure by specific p-value, log 10 of the p-value here, and then an R-squared value if there are any. And then you can also look at both cis acting. This is where the genomic location is on the same chromosome as the probe, or nearby the probe, and then trans acting, where the genomic location is actually affecting the expression of some gene that's not near the genomic location. And then we wanted to, but haven't fully implemented, a way to search and filter by specific GWAS traits or ClinVar traits. So any questions about the filters and what they do? If there aren't any, then I can continue. Just show the initial way this browser works is that all of the studies are loaded on this Studies tab here, and then these are also tied to specific publications. And then if there's individual level data available at various resources, either at dbGAP or the HapMap websites, or even EGA studies, They're shown here. That's where you'd get the individual level data that could be used to recompute the p-values if you wanted to. And then this is for the genotypes, and then for the molecular phenotypes, you can find those data in these resources. And so we have a number of studies that are loaded, and that's what these pages page through. You see that a lot of these are GTEx. So we've loaded for each GTEx tissue, we've loaded that as a separate study. Our initial plan for this browser was to have a tissue tab, and so GTEx would be one study, but it has multiple tissues. But instead, we kind of got a little bit lazy, and we decided that we'd make the GTEx, each tissue within GTEx, a separate study. And so the studies publication lists all the studies. There's 54 loaded now. And like I said, GTEx is really one big study with multiple tissues, but we've loaded those as separate studies. And you can see across here, you can see the number of QTLs that are loaded that are cis-acting or trans-acting for each data set. Cis-acting QTLs are much more abundant usually. And as you scroll across the table, you can see the number of markers that were involved, the number of probes that were involved, the number of subjects that were involved, PubMed ID, and like I said, where to get the data. Any questions about that at this point? Okay, well, if there's no questions about that, then I'll just show you how you might use this, just as an example. Say you were wanted to look at a specific study, and you were interested, say, in the adrenal gland tissue within GTEx, and you wanted to look at drill down on here, and you wanted to filter that further, you could maybe say, oh, actually, I'm quite interested in a very specific gene, and you could just say, so I'm just picking a gene that I know has a low p-value. So when you do a search by the gene, you can specifically pick the study, and then you can display the results. And when you do this, you see that this gene actually has, at this RS, shows a p-value that has very, very low p-value. QTLs are difficult to know what significance is because, genome-wide significance on a GWAS is something like 10 to the minus 7th. The Bonferroni correction on looking at all of the SNPs by all of the genes is going to be something higher than 10 to the minus 7th. I'm not sure what it is. I don't know if anybody does. But the p-value, lower p-value is going to be the more significant hits. And so, any questions about what I just showed? So what we could do is maybe look at what this gene might look like in ClinVar, and we see it turns out that this gene actually is in ClinVar, and it looks like it's related to this Warsaw breakage syndrome. So I'm not sure too much about this disease, but if you were interested in what MedGen had to say about that, you could always look in MedGen and look at that. So I think I didn't do that. But you could look it up, and you could read all about it, understand what this disorder is. So, and then I was trying to come up with an example here that might be relevant to this group. So the other thing I wanted to show you about this browser is that, well, you can download the results or you can display them. the display result just looks like this. Often, I guess, depending on what you've queried here, you might get many results. You can download those if you want to a text file. The other tab here is actually set up where each row is now listed as a bioprocess or a biosystem, so a pathway, as it's understood through the NCBI Biosystems database. And so I had an example that might be kind of interesting to look at here. And here, what we're going to do is we're going to use a different filter. We're going to just look at trans SNPs. And in this case, you're actually looking at a very specific disease that you know. And this is just a gene for the disease that I'm showing. So when I do this, I actually see that there's multiple biosystem pathways that are involved in this gene. And it turns out that there's some trans SNPs that show up across these. Now, these are likely to be similar ones. I could further go back up here and pick which well, I could pick, because there's only a couple, I can just pick one of these pathways that looks maybe interesting to me, and then I can display the results. And then when I do that, I think this is actually a bug in our system because it looks like the same results being shown twice. It does distinguish between these stat descriptions, and I can't comment on that, but it looks like this is false discovery rate, this is slope, standard error. I think because the results are always the same when we do this, this might be kind of an error in our system. But regardless, this shows a p-value of 10 to the minus 8 for this this gene, this pathway, and then you could further look up what that gene looks like in the disease. The interesting thing I think about this is what we're saying here is this RS that actually resides in this gene on chromosome 9 is actually affecting the expression of this gene on chromosome 9, and they're not cis, so it's trans acting. So I think that's my demo, and I figure there's questions, so I'll open it up for questions at this point.
So this is Jenny. So I have just a question about that last. I'm not sure I was understanding the cis and trans. So you're saying that that particular SNP is affecting the expression of that gene because it's in. I'm sorry, I wasn't really following.
It was empirically measured in these
okay.
in the people in this study.
Okay.
That the genotype at this position in the genome.
Mm-hmm.
It depends. People might have an AA or an AT or a TT. I'm not sure what the SNPs are here, the alleles are here, but the genotype at this position in the genome is actually strongly correlated with the expression of this gene that's trans. So it's away from. So that's kind of interesting.
Okay. Yeah.
So
okay. I got it. So it's not within.
Yeah. So we came to it from, we know this person has this disease, and we know that this gene is involved in that disease. Is there some other place in the genome affecting the expression of this gene?
Okay. That's what I was missing. Okay.
And this is it.
Right.
So, okay. Well, and I think there's a question of here, and this is this was actually built as a prototype, and it was built on some older technology, and we're trying to really get some feedback of whether these type of data would be useful, maybe in a more modern form, more updated form. There's also the ability to take values and align them on a genome view that could be embedded within ClinVar or some other resources here at NCBI. You see that we've actually done a number of linking out to, and I didn't show this, but this actually links out to the Biosystems database on this tab, and so you can see the pathway. You could, if you focused in on this, which is a little blurry, I think here, you could see where that gene is involved, and it may be that the other gene where the SNP is not even directly within this pathway, or known to be in this pathway. So, these are the data. I think we're kind of at a point where we're trying to get feedback to decide if this is something we continue, if we want to basically continue developing, or if
Sure.
there's some other view of this that would be useful for people. I'd like to also say that all of these data are available on an FTP site for download. So if you're more of a computational group that just wants to churn through the whole genome, you could download, these, 4 million QTL results and chug through them.
Yeah, that was really cool. And the Biosystems, I hadn't looked at that page before, so that looks really neat too with the different pathways. It's
good to know that, but if you go up here on any, most NCBI sites will have a thing that you can actually pick a specific
Sure.
resource, and if you just do an empty search, it usually goes to the home page, and then from there you can just type in whatever.
So you could put in a particular protein there or gene, or do you have to be more specific?
Well, I think it's just a keyword search.
Okay.
You don't even have to spell it right. So I guess I was lucky because anemia. So it's just a keyword search. So whatever they've indexed for this record,
Okay.
which might be most of it, would come up. If you had a specific example, you could try it. But yeah. So that's what we've done. We've linked out I didn't show it all, but some of the results actually link out to